Good morning everyone. Have a fantastic day. Let us discuss or let us study today the structure of a polynucleotide chain of DNA. You know that DNA is a polymer or chain of deoxyribonucleotides, and nucleotide is the basic unit of a polynucleotide chain. Now, to form a polynucleotide chain, many nucleotides they join or link one another, forming a long chain called as polynucleotide chain. And the bond that joins two nucleotides is called as phosphodiester bond. Very, very important. So name the bond. Question is asked. Name the bond that joins two nucleotides. That is your phosphodiester bond. Clear? This phosphodiester bond is formed by two phosphoester bonds. So phosphoester, that is ester, phosphodiester. Clear? So when the two nucleotides are joining, then one phosphodiester is formed. Clear? Now come to the nucleotide, which is the basic unit that join or link to form your polynucleotide chain. Have a look towards the board, my dear children. A nucleotide, which is the basic unit, consists of three components: one nitrogenous base, one pentose sugar, and one phosphate group make or form a nucleotide. Have a look towards the board, my dear children. I have drawn the two nucleotides. Upper side there is one nucleotide, you see, that is phosphate, sugar and base, one nucleotide. Below there is one nucleotide, phosphate, sugar and base. Now both these nucleotides, these two nucleotides, they link or join through 3,5 phosphodiester linkage. 3,5 phosphodiester linkage. So if two nucleotides are joining, one phosphodiester linkage or bond is formed. Why it is called phosphodiester? Two ester bond, ester, phosphodiester. Clear? Now this phosphodiester bond is forming here in this way. More nucleotides they join or link, forming a long chain that is called your polynucleotide chain of DNA. Clear, my dear children? Do you understand? Now a polynucleotide chain, have a look towards the board, my dear children. This is a long chain where the many nucleotides are joining and finally forming a polynucleotide chain. Poly means many nucleotides are joining. So at one end there will be 5p, means the phosphate is attached to 5 carbon of the sugar molecule, and at the last basal end, that is hydroxyl group is there, that is 3OH, means hydroxyl group is attached to the 3 carbon of the sugar. So these are two ends of the polynucleotide chain. The terminal end has got the phosphate and the basal end has got hydroxyl group. So a polynucleotide chain is formed, and the bond that are joining, that is phosphodiester bond. Here also same thing I have drawn here, see. This is sugar, base and phosphate. One nucleotide, sugar, base and phosphate. Another you see, sugar, base and phosphate. Two nucleotides are joining through a phosphodiester linkage. Are you getting me? Okay now. Now this is the structure of a dinucleotide I have drawn. So a dinucleotide, clear, where one phosphodiester is formed. Two nucleotides means one phosphodiester bond. Three nucleotides, then will be two phosphodiester bond. So number of phosphodiester bond is equal to number of nucleotides minus 1. Remember this thing. For example, if there are 100 nucleotides, then what would be the number of phosphodiester bond? The formula is n minus 1. N stands for number of nucleotides, that is 100 minus 1. So 99 phosphodiester bond would be the answer. It is only for a single polynucleotide chain. Later on you will study double helix structure of the DNA. Here the formula will change. Here it will be n minus 2 because for two strands of polynucleotide chain. Clear? For 1, the formula is n minus 1. Let us now discuss some of the important questions about your polynucleotide chain of DNA. For linking two nucleotides, I have written two nucleotides are linked through 3,5 phosphodiester linkage or phosphodiester bond. What would be the number of phosphodiester bond if the number of ester bond is 100? So it will be 50 will be the answer. Clear? If the number of phosphodiester bond is 100, then what would be the number of ester bond, phosphoester bond? That is 200. Which one form the backbone of the polynucleotide chain? The answer would be sugar phosphate form the backbone of a polynucleotide chain. And a very important thing is that the bases are exposed outside. Have a look towards the board. Bases are exposed outside in a single polynucleotide chain. But when we will talk about the double helix structure of the DNA, the bases, they face towards the inner side. Very, very important, my dear children. Okay, now this is all about the structure of a polynucleotide chain of the DNA.